You may not realize it, but your body is being trained all the time. And over time, those repeated patterns can begin to change what the body does next. Sometimes the body learns to turn a response down. Sometimes it learns to turn a response up. And sometimes it learns that a taste, a pill, or a ritual means something is about to happen. Understanding that matters because it means some of our responses may not be as fixed as they feel. They may be learned, reinforced, interrupted, or reshaped. In this episode, we are going to look at the growing body of research suggesting that biology may be far more history sensitive than we once believed. And if the body can learn from repeated signals, the medicine may eventually become less about repairing the body like hardware and more about changing the patterns it has learned to run. It may become a way of updating the program. One of the simplest ways biology changes with experience is by learning to respond less. Sometimes this is called habituation. Sometimes, especially with repeated chemical exposure or drugs, it is called tolerance. The details are not identical, but the larger pattern is similar. The signal returns, but the body response changes. Biology filters, it adjusts. Variations of this pattern appear across many levels of physiology. In studies of the acoustic startle reflex, researchers found that repeated loud sounds can gradually reduce the body's reflexive blink response over time. In other words, the nervous system begins reacting less strongly to the same repeated signal. Something similar happens in the amygdala, a brain region that helps flag emotionally important information. When people see the same emotional stimulus over and over, the amygdala often responds less strongly over time. The important point is not just that the response fades, it is that living systems are adjusting future behavior based partly on prior exposure. In the 2020 study, optogenetically induced cellular habituation in non-neuronal cells, researchers explored habituation-like behavior. Here, the researchers used human embryonic kidney cells engineered with a light sensitive ion channel called channel rhodopsin. When blue light hit the cells, the channel opened and the cells depolarized, meaning their membrane voltage shifted. And then they stimulated the cells with repeated rhythms of light, ranging from one pulse every 2 seconds to two pulses per second. The voltage response gradually became smaller, and not in a random way either, but it declined in a pattern curve that resembled habituation. The response also depended on the rhythm of stimulation, the intensity of the light, and the cell's prior stimulation history. So the cells were not behaving like identical on off switches. Their response depended on what had already happened to them. Essentially, living systems do not always respond to the same experience the same way twice. And once you realize biology can learn to turn responses down, the next question becomes, can biology also learn to turn responses up? And this is where the story becomes more relatable for some. After repeated irritation, skin can become reactive. After repeated stress, the body can become quicker to respond. After repeated inflammation or injury, the nervous system can become more reactive than it was before. The system becomes easier to activate through a process called sensitization. If habituation is the body learning to respond less, this one is the body learning to respond more. For example, the nervous system contains many inputs that are normally too weak to cross the threshold into pain. With sensitization, though, some of those quiet inputs can be recruited into the pain pathway. The threshold changes. The same signal can produce a stronger response. The body becomes easier to set off. One of the clearest medical studies appears in the paper Central Sensitization: Implications for the Diagnosis and Treatment of Pain. Here, Clifford Wolf describes central sensitization as a prolonged but reversible increase in the excitability and synaptic strength of neurons in pain pathways. In simpler terms, the nervous system becomes easier to trigger and the rules of sensation change. A signal that once felt like pressure may become painful. A painful signal may become amplified. Pain may last longer than the original stimulus, and sensitivity may spread beyond the original site. The key shift is that the body is no longer processing the same input in the same way. It is turning it up. Researchers describe central sensitization as a change in the neurons and circuits that help decide how pain signals are processed. Physiologically, some neurons become more excitable, some synapses become more effective. Inhibition can decrease, and signals that used to be too weak to matter can begin producing a stronger response. And this is where sensitization becomes more than a pain concept. It reveals a broader biological pattern. Sometimes, after repeated or intense experience, a living system becomes more responsive, more watchful, more ready. Now, this can be protective in the short term, but if that amplified state persists, the system can start protecting against threats that are just no longer there. In short, sometimes repeated experience teaches the body, ignore this, and sometimes it teaches the body, pay closer attention next time. And once you see that biology can turn responses down or up, another possibility begins to emerge. Maybe living systems are not only reacting to signals, maybe they are also learning from patterns. Maybe they begin to learn what a signal predicts before the event itself even arrives. Conditioning is when the body learns to connect one signal with another event. A cue that was once neutral, like a taste, room, smell, or ritual, becomes meaningful when it is repeatedly paired with something biological. In simple terms, the body learns. When this happens, that usually comes next. One of the clearest demonstrations came from a 1975 study called Behaviorally Conditioned Immunosuppression by Robert Adder and Nicholas Cohen. They asked a strange question: could the immune system learn a taste? To test this, they gave rats saccharin flavored water, which is basically sweetened water with a distinctive taste. Then they gave them cyclophosphamide, a drug that suppresses immune activity. So the taste and the drug became paired. Later, the rats were given the saccharin flavored water again, but this time, no drug. Then they tested the rats' immune system by giving them something that should normally trigger an antibody response. And the rats with the flavored water made fewer antibodies than the control group, meaning its immune system had been suppressed. The taste itself was not a drug, but after being paired with one, the taste alone seemed to tell the body, turn the immune response down in this case. Here, a taste alone had become a biological signal that acted like a drug. In cancer treatment, researchers studying chemotherapy associated nausea noticed something else. Some patients could begin feeling sick even before treatment started. They found the clinic room could matter, that smell could matter, the treatment ritual itself could matter. Over repeated treatment cycles, the body could begin linking the environment with what usually came next. And in some cases, clinicians found they had to vary aspects of the treatment context because of the patient's anticipatory physiological response. And a similar idea appears in addiction research as well. In the paper titled Heroin Overdose Death: Contribution of Drug Associated Environmental Cues, Shepard Siegel explored an interesting possibility. A drug tolerance may not depend only on the drug itself. It may also depend on the setting where the drug is usually taken. When the body receives the same drug again and again in the same environment, it becomes part of the pattern. The body may begin preparing for the drug even before the drug fully takes effect. In Siegel's animal experiments, rats repeatedly received heroin in a particular environment. Later, when they were given a much higher dose, they were more likely to survive if they received it in the same familiar place. But when that high dose was given in a different environment, survival dropped. That suggests the familiar setting may have helped trigger a learned compensatory response. Here we saw a taste can become immunological, a medical environment can become physiological, and a room can become pharmacological. And once you start seeing these studies side by side, a deeper question begins to appear. How is the body doing this? How does repeated experience become embedded strongly enough that physiology itself begins changing before the event even happens? This is where the paper Cellular Signaling Pathways as Plastic Protocognitive Systems becomes important. Matthews, Chang, Devlin, and Levin are not arguing that the body works like a simple machine. They are arguing almost the opposite. A lot of medicine treats biological pathways like wiring diagrams. This receptor activates that protein changes that gene, this drug blocks that target. Now, this model is useful, but the authors argue that pathways may also have to be understood as trainable systems. A pathway does not exist alone in the body. It sits inside a cell, inside a tissue, inside an organ, inside a body, and lastly, inside an environment. So the same molecular signal may not always produce the same result. It can depend on the system's history. It can depend on its current state. It can depend on signals arriving from other levels of the body. And that is what they mean by looking at pathways as plastic and protocognitive. Here, a system can receive information, respond to it, change from it, and carry some trace of that into the future. In the paper, they point to protein pathways and transcriptional networks that may show trainability, memory like behavior, and context sensitive information processing. One of the more concrete examples they discuss is gene regulatory networks. A gene regulatory network is a set of genes and signals that influence each other's activity. The interesting idea is that the memory does not have to live inside one molecule, it can belong to a network state. Some models suggest that biological gene regulatory networks can store multiple memory like states, resist noise, and even perform simple computational tasks, such as counting discrete stimulus events. This can result in a pattern of response, a state the system can enter, a way the past input changes what the next input does, essentially, an update to the software. And the paper extends this beyond chemistry and into bioelectricity because cells do not only communicate through molecules, they use ion channels, membrane voltage, and gap junctions to form electrical networks across tissues. Matthews and Levin describe these non-neural bioelectric networks as another place where memory, context sensitivity, and information processing may occur. It can be partly carried by gene networks, protein pathways, electrical gradients, feedback loops, and tissue states. So the body is not always starting from zero, it has a history. And if a living system can hold a history, settle into patterns, and respond differently depending on its state, the medicine may eventually do more than repair hardware. It may learn how to change the inputs, timing, context to help shift the body into a different state. So, what does all of this actually change? Maybe it changes what we pay attention to. If biology is history sensitive, then the patterns around us are not just background. The routines we repeat, the rooms we return to, the cues we pair with stress, relief, pain, medicine, sleep, food, or safety. All of these may be part of what the body learns to expect. That does not make the body weak, it makes the body adaptive. It is constantly asking, what usually happens here? What usually comes next? And maybe this is why small repeated things can matter more than they seem. A ritual before sleep, a morning sequence that tells the system it is time to wake, a repeated environment that teaches the body to tense before anything has actually happened. They are signals. And if signals can train physiology, then paying attention to our repeated signals becomes a very different kind of health question. How to disrupt patterns that no longer serve us, how to reinforce patterns that help restore function, how to speak to living systems through timing, context, signals, and state. So, can we reprogram the body? In some ways, the answer appears to be yes. Not completely, not simply, but enough to take the idea seriously. Because the evidence points to something profound. The body is adaptive, trainable, listening to repetition, quietly carrying the history of what it has learned. I really cannot wait to hear your thoughts on this topic and let me know if you have had any experiences regarding this. Until next time, stay curious and take care.